Imagine if you could turn an ordinary body cell into a pluripotent stem cell, simply by stressing it. No genetic engineering, no complicated reprogramming factors, just a brief exposure to an acidic environment. In 2014, Japanese researchers claimed they had discovered exactly that. They called it STAP, Stimulus-Triggered Acquisition of Pluripotency. And if the results were real, they could have fundamentally changed how we create stem cells. But within months, the discovery began to unravel. To understand why STAP was so extraordinary, we first need to understand what pluripotency means. A pluripotent cell can give rise to almost every cell type in the body. Embryonic stem cells have this ability naturally. Scientists can also create induced pluripotent stem cells, or iPSCs, by introducing specific transcription factors that reset a differentiated cell into a pluripotent state. But this process is relatively complex. The STAP experiments appeared to offer something radically simpler. Researchers took differentiated cells from newborn mice and exposed them to a low pH environment. The cells experienced a strong stress, and according to the researchers, some of them responded in a remarkable way. They began expressing OCT4, one of the key genes associated with pluripotency. The researchers used mice engineered to produce GFP fluorescence when OCT4 was activated, allowing them to identify these cells under a microscope. The apparent transformation was striking. Ordinary somatic cells seem to be acquiring characteristics of pluripotent stem cells. But simply turning on OCT4 isn't enough to prove that a cell has become pluripotent. So the researchers performed additional experiments. They reported that STAP cells underwent extensive epigenetic changes around genes, such as OCT4 and NANOG, consistent with a more pluripotent state. They also performed differentiation experiments. The cells could reportedly produce derivatives representing the three germ layers: ectoderm, mesoderm, and endoderm. And when the cells were transplanted into mice, they formed teratomas containing multiple tissue types. These were all important pieces of evidence. But there was an even stronger test. If a cell is truly pluripotent, it should be able to contribute to the development of an entire organism. So the researchers injected STAP cell clusters into mouse blastocysts. The resulting embryos developed into chimeric mice. Cells derived from the supposed STAP cells were reportedly found throughout the animal's tissues. And the story went even further. The researchers reported germline transmission. In other words, genetic material originating from the STAP cells could be passed on to the next generation. They even reported results from tetraploid complementation, one of the most stringent tests of developmental potency. In this experiment, STAP cells were reportedly capable of generating essentially the entire embryo. If all of this was correct, the implications were enormous. A mature cell, exposed to a simple physical stress, could apparently return to a pluripotent state. But shortly after the papers were published in Nature, researchers began noticing problems. Some figures appeared suspicious. Some images appeared to have been manipulated or reused. And questions were raised about the experimental methods and data. Riken, the institution where the research was conducted, launched an investigation. The investigation examined several specific allegations, and the findings were serious. One of the problems involved a figure showing T-cell receptor gene rearrangement. The figure was presented as evidence supporting the identity and origin of the cells, but investigators discovered that it was actually a composite image created from two different electrophorest gels. One lane had been taken from a separate gel. The image had also been vertically stretched and contrast adjusted before being inserted into the figure. According to the investigation, the lane had not been positioned using scientifically appropriate criteria. Instead, it had been visually aligned with the neighboring band pattern. The committee concluded that this constituted research misconduct through falsification, and this was not simply a formatting error. The manipulated image could give the impression that results from two separate experiments had come from the same gel. Another problem involved images supposedly showing differentiated STAP cells and teratomas. Investigators discovered that several images closely resembled images from Haruko Obokata's doctoral dissertation. The problem was that the experiments represented by those images were not the same. The Nature paper described cells generated using acid treatment. The doctoral work involved cells generated under mechanical stress, yet images from the earlier work had appeared in the STAP paper. The investigation concluded that this was research misconduct involving fabrication, and because these images were important evidence for the claimed pluripotency of STAP cells, the committee stated that their use seriously undermined the credibility of the data. Finding falsified or fabricated data does not automatically tell us whether the underlying biological phenomenon is impossible. Maybe the experiments contained serious mistakes, but perhaps STAP cells were still real. So the next question was much more straightforward. Could anyone actually reproduce the phenomenon? And eventually, Riken attempted to find that out. Haruko Obokata herself participated in an attempt to reproduce the STAP phenomenon. Researchers used the same general idea: cells from newborn mice, an OCT4 GFP reporter, and low pH treatment. After several days of culture, GFP-positive cell aggregates could indeed be observed. At first, this might sound promising, but there was a major problem. The frequency of these GFP-positive aggregates was approximately 10 times lower than what had been reported in the original studies. GFP fluorescence alone does not prove that a cell has become pluripotent. The researchers noted that fluorescence could potentially be influenced by autofluorescence or nonspecific expression associated with stressed or dying cells. So they looked more closely. Most of the aggregates did not convincingly express the expected pluripotency markers. And then came the most important experiment. The researchers took the putative STAP cell aggregates and attempted to test their developmental potency. They injected these cell clusters into mouse embryos. In total, 1,051 embryos received injected cell material. Of those, 591 embryos were recovered and examined. And the result? None showed significant contribution from the injected cells. No convincing chimeric contribution. The key developmental property reported in the original STAP papers could not be reproduced, and importantly, the researchers acknowledged limitations. The experimental conditions could not be guaranteed to be absolutely identical to those used in the original experiments. So this experiment alone could not prove that every possible version of the STAP protocol was impossible, but it did demonstrate something extremely important. Under these conditions, even the original researcher failed to reproduce the defining developmental result of STAP. As the problems accumulated, confidence in the original findings collapsed. The investigation concluded that 2 instances of research misconduct had occurred, and eventually, the 2 STAP papers published in Nature were retracted. The extraordinary claim had failed the most important test in experimental science. Could the result withstand independent scrutiny and be reproduced? The STAP story is sometimes presented simply as a story about scientific fraud, but there's a deeper lesson here. Science doesn't depend on scientists being infallible. Scientists make mistakes. Experiments fail. Unexpected results happen. What makes science powerful is that extraordinary claims can be tested, challenged, reproduced, and corrected. In science, a discovery isn't established by how extraordinary the result sounds. It's established by whether the evidence survives scrutiny. Thank you for watching.